Well, welcome this afternoon to our NextGen and Ellis Michelle Cancer Lecture. It's my pleasure today to introduce Dr. Barry Edwards, who is currently an associate professor here in biochemistry, as well as radiology. He received his Bachelor of Science degree in chemistry in 1985 from the University of North Carolina in Asheville. It's a beautiful place. During 85 and 86, Dr. Edwards studied at the Philipps Universität in Marburg, Germany as a Fulbright scholar, and he received his Master's of Science in Organic Chemistry from Florida State University in 1990. He worked in the laboratory of Michael J. Welch from 1990 to 1994. Then he joined the graduate program in bioorganic chemistry at Washington University in St. Louis and completed his PhD under the direction of Dr. Welch in 99. His thesis project was the evaluation of radio-labeled peptides for imaging integrin receptors. In 2000, he joined the Metaphor Pharmaceuticals in St. Louis, Missouri, where he developed chelators for iron overload disease and manganese superoxide dismutase mimics for pharmaceuticals for pain relief. In 2005, he joined the laboratory of Samuel Achillefu as a research instructor at Washington University in optical imaging, and he was promoted to assistant professor in 2009. Then in 2011, he joined the radiology department at the University of Pittsburgh. And in 2020, he was recruited here to the University of Missouri, where he holds joint appointments in biochemistry, chemistry, and radiology as an associate professor. His research interests include development of peptide and protein-based probes for theranostics of cancer. And I know he has some new information to share with us today. And so thank you, Dr. Edwards, for joining us and lecturing to us today.
All right. Well, thank you, Dr. Brian, for that fine introduction. A little walk down memory lane. Forgotten a lot of that stuff. You go out and look on the internet now for Metaphor Pharmaceuticals, you might find a teeny little wisp of information, but it was good that they laid me off because it got me back into doing what I really love doing. That's molecular imaging. Love talking about it, and I love getting other people doing it too. So that might happen today after I give this talk. So, all right, so let's get started. So first we'll have a little overview. And we're starting out with just the basics of PET, molecular imaging, and then we'll get into some of the projects I'm going to present. ImmunoPET of TAMCs with anti-CD11 antibodies. And then we moved on then to target those TAMCs for a theranostics project with a new chelator we got from, or we had in collaboration with a company, Lumifor. And then finally, hot off the presses, some new things that we've been doing to try and image activated T cells in the tumor microenvironment. All right, so just the basics of nuclear imaging, you always have to have some contrast agent, small molecule, peptide, polypeptide, the list goes on and on, DNA or RNA, but that contrast agent is labeled with a radionuclide, and that's injected IV for circulation and distribution through the tissues. And so we are able then to understand, and especially when we target saturated molecules like receptors or enzymes, we can understand functions of tissues because we can quantify those molecules that are upregulated as a function of disease. There is an ionizing radiation involved, but it's generally relatively safe, and probably more radiation when you get the CT. That always goes along with the PET. Yeah, so here's a modern PET scanner here. You can see the patient slid through those rings, and that there. I don't think these days it's impossible to buy a PET without accompanying CT. A couple images here over on the right. You see a CT image, and there's a PET image right next to it. And I'm pretty sure this is FDG because the brain is lit up. You can see a little bit of bladder there, and this is a normal patient. You don't see any hot spots, but it's really then the co-registration of anatomy with PET that really is powerful in understanding the nature of the disease. So we're working on antibodies and antibody-based, and at the core of that approach, one needs a bifunctional chelator for preparing these molecules. And one function of the chelator is to hold that radiometal and not let it go. The second function is a sturdy and stable covalent linkage to the targeting molecule. Now, some properties that are desirable in developing one of these chelators, it needs to react with the radiometal quickly. And in a case of proteins like IgG, you wouldn't want to have to heat it too much. And especially in the case of IgG, antibodies, you don't want any disulfide bond reducing agents because those bonds are exposed, solvent exposed in IgG. And of course, it has to be stable. So when those conditions are met, you can then go on and optimize target tissue uptake and then minimize non-target uptake. And here we're just a little cartoon to show you what the whole complex looks like. You have a targeting group, the binding, some binding domain for a cell surface receptor, and then the radionuclide holding on to the radiometal, decays, beta decay, and then combines with electron for annihilation. And you have the two 511 keVs 180 degrees apart, and that really constitutes the basis of PET. Some of the radionuclides that we're interested in right now are for antibodies are copper 64 and zirconium 89 due to their half-lives. You have to match the half-life to the circulation properties of the molecule. Now, gallium-68 is really more in the province of peptides or some very, very low molecular weight antibody fragments with that 1 hour half-life. Those copper-64 and zirconium-89 are easily prepared on medical cyclotrons. And we can get both of those every week, either from Washington University or the University of Wisconsin up in Madison, Wisconsin. So there's no problem in attaining those radionuclides. Here's this is the core technology we've been using to radiolabel our IgG molecules. And we start with a readily commercially available chelator called DFO, dysferrooxamine. And it's a great key. And reaching back in time, back in metaphor, we're looking at orally bioavailable alternatives to DFO for iron overload disease. That was my first experience with this molecule, but it also just happens to be a great chelator for zirconium-89. It's readily conjugated to epsilon amino groups of lysine on the antibody. And we really usually try not to do excess chelation. That could cause problems. So we usually shoot for about one up to 3 chelators per antibody. And there's a number of ways to determine your substitution level. And right now, we found a UV method that's pretty reliable. We can work that out by size exclusion HPLC. But it's very important to get rid of that excess chelator before you do a radiolabeling experiment, because we've been burned that way before, and I'm always on the trainees to make sure they get rid of it. And usually use some size exclusion method, size exclusion HPLC or some centrifugal filters. They're pretty effective. I'm often asked when I give lay talk like this, so how do you keep from, destroying your antibody affinity? And the answer is there's a lot of lysine residues that are solvent exposed on the antibody. Now, some are more reactive than others, but before I saw this paper by Gaudier, I thought, well, we're doing a conjugation. We might have, we're choosing from all 80, but the truth is at a certain concentration, those authors showed that when you set a concentration of your agent, in this case, they looked at NHS esters. They're able to show at that, at one single concentration, you only got about a dozen sites conjugated. And if you wanted to get more, those are the red ones. So if you wanted to get more conjugation sites, then you go on up and you just have to use a higher concentration till you get right on up to some ridiculous levels to get those less reactive ones. So the truth is we're probably just targeting, when we do a conjugation, very, very few of those lysine residues. And just some notes. I mean, you don't, the worst thing you could do is destabilize your antibody. The first worst thing is you could lose affinity. but that we really usually don't see that. Well, but the concern is that we might cause a destabilization and aggregation, and then your antibody starts precipitating. Now, I couldn't talk about doing a molecular imaging experiment, a talk, without talking about affinity, because it's come up so many times over the years when collaborators have come around saying, hey, let's try an experiment. And that's usually the first question we ask is, what's your affinity? And how does it affect tumor accumulation? Well, this very, very oft-cited papers by Adams in Cancer Research, what the authors showed is that when you get up to about 1 nanomolar, affinity doesn't improve any. So there's no need to try to get affinity up to the highest level possible. In this case, these were scFv antibodies, and that's the smallest functional fragment of an antibody. It's just VH and VL tethered together with a 15 amino acid linker. And here they were targeting a HER2 receptor. So, and that's been shown many, many times. And the fact for IgG, even KD values up around 100, 200 nanomolar are sufficient for good tumor uptake. Part of that is for the reason, especially for IgG, is their serum circulation times are so long. And that plays an important role in tumor uptake. Now, I'll spend a little time talking about molar activity. Now, we radiopharmaceutical chemists think about this a lot. So, high molar activity is a case where there's a high ratio of labeled antibodies to unlabeled antibodies. Now, this little cartoon I made here is an artist's conception. The truth is that in a radiolabeling experiment, there might be 1/100th of the molecules might be radiolabeled, or even less, 1/500. After extravasation and receptor binding, the outcome of high molar activity is a fair amount of receptor occupancy of the radiolabeled antibody or whatever ligand. And that leads to good sensitivity in the PET scanner. If we contrast that to low molar activity, a low ratio of labeled antibody to unlabeled antibody, after extravasation and receptor binding, there are very few labeled there. Now, we go to those extremes sometimes when we're wanting to show specificity of our ligand for the receptor. We basically increase the molar activity to the point that none of the tracer binds, and that's competitive inhibition, and that can be very valuable. But sometimes, though, we're stuck in a case where we have a lot of antigen sink of having a problem, and I'll get to that right now, the consequences of antigen sink. So in any radiolabeling experiment, we have target tissue and usually tumor with upregulated receptors. And we have also antigen sink, which is a reservoir of receptors in off-target tissues. And the net result of that is that can deplete the tracer accumulation in the target tissue. And this is especially started to come up for us when we're doing imaging cells in the tumor microenvironment. Blood, for example, can be a significant antigen sink. So the solution to this problem is some way and somehow to block that antigen sink with unlabeled antibody. And that can be pre-administration of the unlabeled ligand, optimization of molar activity, the experiment just devoted to that, and then or some variation between the two. All right, let's discuss one of the first projects I did with my collaborator. I'll introduce him in a minute. We looked at ImmunoPET of TAMCs in the tumor microenvironment of a mouse model of glioma with an anti-CD11B antibody we labeled as with zirconium-89. Now, I was fortunate enough to meet Gary when I was up at Pitt, and I'd always wanted to work in the field of glioma because I just felt, imaging would be so important in that field. And so together with Gary and his group and my lab member, Shivanshi Nigam, which has gone on to a career in industry, we tackled the problem. So first, a few notes about pediatric cancer is that brain and CNS tumors are one of the most common solid tumors and cancers in children. And they're also sadly the most common cause of death in children. And they tend to occur in the sensitive region of the infratentorial region. Now, why we're interested in those TAMCs, MDSCs, and myeloid-derived suppressor cells and tumor-associated macrophages is that they represent an immunosuppressive pool of cells that hinder natural tumor immunity. And knowing that, then we should be able to stratify patients then that would benefit from and there are some therapies out there for to knock down that pool. So it'd be important to identify those patients with molecular imaging prior to therapy or during therapy to monitor how it's doing. TAMCs are compose a huge part of gliomas, especially glioblastoma. And yeah, there are multiple trials targeting those TAMCs, and there was a clinical trial a few years ago, and it did not do well. But as far as we know, no one's doing PET imaging or has done PET imaging for these types of cells and tumors. So let's see how it turned out. So we started with an anti-CD11B IgG. It's a pretty well-known clone, the M170 clone. And it just so happens to bind mouse and human. So we'd have a shot at it'd be a much easier shot to translate this molecule or fragments thereof just because of that right there. So we start with a well-known mouse glioma, GL261, injected into the right hemisphere. DFO for our chelator. We always do MRI to evaluate tumor size prior to embarking on one of these experiments and make sure that the tumors are relatively uniform, and then we don't use mice where tumors are undersized or oversized. We'll have some histology of the CD11B tumors to show in a moment. And we also did flow cytometry of those tumors, and looks like I've got a result here already. In that we found there was about 50,000 receptors per cell on those TAMCs collectively. And that's an important result because too often in our field, it's hard, or no one really goes to the trouble to figure out what the receptor density is. And that plays a major role in what kind of sensitivity that ultimately you see. So here's the results, 72 hours post-injection. You can see in that top row there, strong uptake in the tumor in the right hemisphere. And then blocking happened to work really well here. We just reduced these molar activity tenfold, and that was enough to completely abrogate binding of the receptor. And over on the right, you can see the biodistribution results. And if you're not familiar with that, that's just tissue counting. And we represent those results in terms of percent of the injected dose per gram of tissue. Now, the reason we do that is that the tumors themselves, they're all a little bit of different sizes, and we normalize it to the weight of the tissue. And the other reason we do that is that if you don't normalize to the weight of the tissue, the big organs get all the radioactivity, and your brain, your mind's left to sort out teeny little differences from the other organs. And by normalizing it to weight, then high uptake in a case like we're seeing here, I think the average was about 8 or 9% ID per gram, and it makes it really stand out. Now we are seeing a little bit of uptake over in the contralateral side, and it's definitely much less than we see in the right hemisphere. Now we counted both the hemispheres. Now we didn't dissect the tumor. And the reviewers asked us to do normal brain, and you see there's just next to nothing in normal brain. Way down here, about 0, just barely detectable. So we don't quite know what's going on at the blood-brain barrier on the contralateral side. Now, for certain, blood-brain barrier is disrupted in GL261s. That's well known. And we don't quite know what's going on over on the contralateral side, whether there's little bits of tumor sneaking over there that we don't see in PET, or if we somehow actually altered the permeability in the contralateral side, because there is natural CD11B in the microglia. For a control there, we've got some immunohistochemistry. You can see really strong staining in the tumor, very low staining in the brain. And we were able to do a little bit of quantification, and you can definitely that we got stronger staining in the tumor than we do in the brain, and it was statistically significant. And there's an MRI that we performed to follow tumor growth. All right, so summary then of that work, then we were able to clearly visualize those TAMCs in that animal model. And these results, we believe, demonstrate translational potential for this type of work to quantify TAMCs in the tumor microenvironment. And this could be used for determination of how many TAMCs there before, during, after therapy, deciding whether to embark on immunotherapy, because that's an expensive treatment and not without side effects. All right, so moving on then, we'll having done that, we wanted to move into developing a theranostic to not only visualize TAMCs, but also target them with targeted radiotherapy. So in this one, we teamed up with some scientists from Lumiphore that had developed a new chelator, and Alex Foster in Gary's lab and Shibachi, Nagam, and my lab worked on this. And we also teamed up with Carol Anderson, who's an expert in targeted radiotherapy and also the chemistry of radiomelts. So the basics of a theranostic are molecules that can see or treat disease. And just like a simple imaging agent, they can be just about anything that can carry the isotope, both isotopes, and there's a number of nanoparticles that can function this way. Of course, nanoparticles can carry just about anything. Small molecules can also function as theranostics, but we'll get into how we used an antibody as a theranostic. So for PET imaging, of course, we're going to utilize zirconium-89 as our radionuclide. And for our therapeutic radionuclide, we chose lutetium-177. Has about a half-life of 6.7 days, so that's good. So if you can get your radionuclide in a tumor, it's got a long time to blast it with beta particles, and that can increase your chances of therapeutic success. Now, it does have some gamma emissions, but they're a little on the high side, and those could be used for SPECT. And but the images have never really been that great with lutetium-177. So that it's really for that reason you want to have this therapeutic pair. I mean this theranostic pair of isotope, one for imaging and one for therapy. So some of the questions and challenges that we faced when we were doing this is that if we actually could target and reduce those TAMCs, would that improve survival? And what chelator we can use? DOTA, it's a great chelator for lutetium-177, but not for zirconium-89. And DFO is a chelator we always use for zirconium-89, but it does not complex lutetium-177. So that's where Lumiphore came in, because they had a chelator that they felt would be perfect for both of those isotopes and would promote very facile radiolabel. So yeah, there really wasn't a lot of chelators at the time that could really perform this dual role. And that's where the molecule or the chelator Lumi-804 came in, because we could label both zirconium-89 or label an antibody with both those radionuclides at room temperature relatively fast. And here's a few crystal structures here if we have any chemistry folks looking at and are interested in that. And there's the structure of Lumi-804 over there on the left. A little chemistry here. They provided us that Lumi-804 is a calcium complex. And the reason they did that is that sort of protected those N-hydroxy compounds so they could prepare the NHS ester. That's how we coupled it to the epsilon amino groups. And then after that, it was very facile radiolabeled, both radionuclides at room temperature, really fast, no heating involved, high radiochemical yields. You can see that on the rightmost panels. No aggregation induced. The affinity of the antibody after conjugation was very high, was not compromised. And we did a serum stability study where we took both zirconium-89 and lutetium-177 labeled antibodies and stewed those up in mouse serum. And we didn't see any dissociation of the radiometals over that time period. So with that, then we were ready to go in vivo and try our experiment, our imaging and radiotherapy experiments. Here's the first results with lutetium-177. And you can see over here on the left the SPECT imaging, and the tumors do show up, but the sensitivity isn't really very good. And we had to count for quite a while. I think we had those animals in the scanner for an hour, even 2 hours, just to get these images here. But let's focus our attention on the biodistribution. We do see high uptake in the ipsilateral region. That's the right hemisphere where the tumor is relative to the contralateral on the other side. Another important feature was, and it's a little inexplicable, is that the blood concentration was extremely low. So we never really fully understand that, but that's the result we got. And then other features too, there's always high spleen accumulation. That's a reservoir for CD11B. And we see some thymus uptake too. And now thymus is a teeny organ, but seeing that though is always extra information to us that our molecule is functional in vivo. All right, so in the PET-CT, you can see as before, as we did with DFO, we got very good accumulation in the tumors. There's a pair of transaxial images there on the left, and we're able to block that down with excess CD11B. That proves that we're receptor specific. In the middle panel there's some SUV values, and that's basically just the unitless value. And I'll have it defined later on in the talk, but it's just the quantity of the percent ID per gram divided yet again by the mouse weight. So statistical significance between the ipsilateral and contralateral sides, very low uptake in the contralateral side. Let's see what else we have here. Now the blood's a little bit high in this experiment, and we're not really, again, the spleen's probably releasing CD11B positive cells, and we could block that down. So there are a few other sites where we could block receptor accumulation. The liver, let's see, lungs, and was one place that we could get statistical significance. So again, sometimes the blocking never really, when you have these pockets of antigen sink and you go off trying to block, you can knock tracer out of one compartment and send it off to the other. So sometimes it really makes the results difficult to understand, but at least we were able to block the receptor accumulation in the tumor. So then we went on to try some therapy experiments with lutetium-177, targeted radiotherapy experiments. So we started with 7-day-old GL261 orthotopic tumors, a couple of doses, one single dose for about 15 or 22 megabecs. And then 72 hours after we injected them, we euthanized those mice, and we did some flow cytometry in the spleen and the tumor, and antibody staining, and to understand what was going on with these. And what we observed was that CD11, the targeted radiotherapy in the spleen decreases the MDSCs and the macrophages, but it did not decrease the granulocytes or, importantly, the T cell populations in the spleen. So that's a good result because really we're ultimately trying to not interfere with natural tumor immunity. And the spleen's a reservoir for cells that can be released and then traffic back up to the tumor site. Now in the brain, we were only able to decrease MDSCs at the higher dose, and we did not affect the quantity of the tumor-associated macrophages. Granulocytes, again, were not changed, and again, no changes in the tumor-infiltrating lymphocytes in those cells, those CD3 positive lymphocytes that are either CD8 or CD4. So what we decided to do then is to try to fractionate the dose and see if we could get a better result, because there was some toxicity at the higher doses, and we couldn't see any survival benefit at those higher doses. So up there in the upper panel, you can see how we fractionated the doses out, and we included checkpoint immunotherapy, ICI, with both an anti-PD-1 antibody and an anti-CTLA-4 antibody. And at the very end, we did MRIs for tumor volume, and we looked for symptom-free survival as our readout. And we did see the combination of targeted radiotherapy and immune checkpoint immunotherapy was more effective than either of those treatments alone. So then in summary then, the TAMCs are a targeted pool of immunosuppressive cells. And so understanding the quantity of those would be important in directing therapy. We validated this new chelator, LUMI-804, and showed that it was very facile radiolabeling with both of those radionuclides. And they were mild conditions, so easily suitable for antibodies. And the diagnostic part of the theranostic worked really well. We could readily visualize those TAMCs in the mouse model. And we did show some promising results of targeted radiotherapy with lutetium, LUMI-804, anti-CD11B. So I think then with this chelator validation, then that it can serve as a cornerstone for any type of antibody-based theranostic with both the zirconium-89 and lutetium-177. Let's talk about activated T cells, for example, and how could we image those? Those cytotoxic T cells are really what's going to kill the tumor cell, and if you could ever get those activated, then it could be really powerful in anti-tumor therapy. So currently the early approaches were really in imaging CD8 positive T cells. And the reason for that is that the quantities are generally higher in tumors and are associated with better outcomes and overall survival. That's pretty well known. And that was where the initial ImmunoPET started. And as a matter of fact, a company called Imaginab has got an anti-CD8 antibody fragment in antibody in clinical trials right now. But the shortcoming of this is that there's another pool of CD8 T cells in the tumor that are just, they're bystander cells. And so it's, they make up a pool of cells that aren't doing anything. And that could really compromise an understanding of T cell activation within the tumor. So we knew we wanted to move on and select some tumors that or some markers for activated T cells that might be more representative of tumor activation. So I think I promised some slides on immune checkpoint immunotherapy, and here's a couple from old cancer.gov. So everybody's on the same page, because that's going to be important in coming up. In our experiments that I'm getting ready to show, we used an anti-PD-1 antibody to promote or to make sure that we had activated T cells. So in the leftmost panel, when a T cell binds a tumor cell for putative for killing, if that tumor cell expresses PD-L1, its cognate receptor on the T cell, PD-1, will bind it, and then that T cell is shut down, and it has no killing properties at all. So the solution to that is to use blocking antibodies. In the cartoon there on the right, either an anti-PD-L1 antibody or an anti-PD-1 antibody then sterically hinder that interaction, and then the T cell can go about its business, and you can have tumor cell death. We administered anti-PD-1 antibodies. We also administered anti-CTLA-4 antibodies. So again, the binding of CTLA-4 with its cognate receptor B7 can inhibit activation of T cells. So again, by using an anti-CTLA-4 antibody, that process is blocked, the T cell can become active and stay active. And again, the result of that is, again, cell killing from cytotoxic T cells. This is one of my favorite videos. I watch this one a lot, scientific videos, that is. And this is a video that was done with intravital microscopy. There's a lot of lessons to take away from this one. So what the authors did was they had an MC38 tumor they implanted in dorsal fold in the skin in a mouse. And the movie I'm getting ready to show is just before administration of an anti-PD-1 antibody with a dye on AF647. They pseudo-colored that one white. So what we have here are tumor cells red, tumor-associated macrophages blue, T cells green. So here we go. Now what you can see right now, let me pause it. Crap. Doesn't look like it's wanting to pause. Here. So let me start it again. You see the T cells moving around. So the idea that I think here is you don't need a lot of activated T cells to be effective. All right, so there's the anti-PD-1 antibody appearing in the vein, extravasates. You can see co-localization of PD-1 on the T cells. So a lot to take away from it there, but it's a good visual of what's going on inside the tumor. What target should we select for imaging activated T cells? And what we thought to select was CD69. It's in adult gliomas, it's highly expressed on the TILs, tumor infiltrating lymphocytes, relative to peripheral blood monocytes in patients. And in this data over in the left panel, this is tumor RNA-seq data from pediatric patients, and CD69 was a molecule that stood out as having upregulated, or that was upregulated. So we decided to focus on that one and see how the results turned out. So we use DFO for radio labeling. Over here on the left panels, some overlaid chromatograms, size exclusion HPLC. We see a nice single peak consistent with a molecular weight of a DFO labeled anti-69 antibody. And then overlaid on that is a radiochromatogram, the one that's kind of jiggly, if you will. That's the radioactive peak, and it elutes later just because the UV and the radio detector are about a minute or so apart, a couple of minutes. And there's also mass associated with that. We can see that there in that light gray peak. That's just, now it's marked zirconium-89, but it is not. We're not going to be able to see that teeny amount of zirconium labeled antibody. But the point is that that peak shape looks the same as the antibody before we radio label it. But in the radio labeled antibody, you see a little shoulder ahead of that, and we don't quite know what that is. It suggests there might be an aggregation going on there, because that's usually how aggregation looks. So I report it, we don't know. These are the things radiopharmaceutical chemists worry about when we start formulating our molecules. We had high radiochemical yield. We selected a middle-ish kind of molar activity there, 300 microcuries per nanomole. That's just based on experience. And we also performed what was called an immunoreactivity experiment. In that type of experiment, you just determine the bindable fraction. You ask the question at excess antigen, 100-fold excess antigen to the radiolabeled antibody, how much is going to bind. And this one turned out to be pretty high. And it also speaks to high affinity too, because if we didn't have high affinity, all those rinses, I don't think we would have gotten that kind of immunoreactivity. All right, here is the outline for the animal model and the ICI dosing. Again, GL261s in the right hemisphere. We had ICI treatment with both antibodies, 200 micrograms per dose injected intraperitoneally. And I would like to note that this is a subcurative dose. You can cure mice with GL261 with the combination of anti-PD-1 and anti-CTLA4 treatments. We tried a blocking experiment, and you'll see the results of that in just a moment. We performed PET-CT imaging at 24, 48, 72, 96 hours, and one last time point, 144 hours. And at that time point, we did a biodistribution. And here it is. We did not see any differences between the checkpoint immunotherapy group and the block group. So those were combined without consulting a statistician. Now we may change our minds and undo that, but for the purposes of today, that's what we're going with. And you over on the in the right brain, you can clearly see that we can block down the binding of the zirconium-89, anti-CD69 tracer, telling us that our accumulation in the tumor is receptor mediated. Now, some other salient features is that there's a fair amount of blood lingering even at 144 hours. And that gives us a relatively low tumor to blood ratio. And this is one of our key benchmarks for performance. And in our case, it's definitely less than 1. So it leaves, we do definitely see receptor mediated accumulation. So we'll look at some more of our experiments to try and understand what's going on here. So here's some of the PET-CT images. Even as time goes by, definitely see greater accumulation in the tumor. In the top row, the untreated control group accumulation is very low relative to the checkpoint immunotherapy group down in the bottom row. Over on the right, you see what we call maximum intensity projections. So this is the full image of these mice, and a lot of uptake in there in the liver and in thymus that is not in the PBS treated group. Now, in the spirit of full disclosure, I talked to the trainees that put this together, and they said, "Well, Dr. Edwards, we picked the best looking ones." So if we go back up here, some of those mice, if we looked at all of them, I don't know that they would look that good. So, but this is for publication, so that trainees pick the best ones. So we did, one of the group members, Michal, she did a fair amount of flow cytometry. And so in those experiments, there was the treatment schedules down below in the lower part, ICI treatment at days 10 and 12. And then after that, the tumors and the spleens were removed for a flow cytometric analysis. Now up above in purple, I've shown how that corresponds to our imaging results. So here's some of the flow cytometry from the tumor. There's definitely an increase in CD69, CD8 positive, and CD4 positive TILs in those tumors, and there's a fair amount of variability in some cases as time goes by. I don't know if that totally corresponds to the PET data, but these are the results that we have now. And another experiment too, and she also did spleens. You can see there's a little bit of a mismatch in timing when so the left axis is the CD69 expression ratio relative to the pre-treated group. See, at 2 days, the CD8s in the tumors, those TILs are a lot higher, and then it's only 2 days later than they are in the spleen. And so I think this was a surprising result for them. I wish I could discuss it in a little bit greater detail, but that was the observed result. But the bottom line here is we do see increases in CD69 in the spleen and in the tumor. So with all this up and down, all these numbers we had, the question that was asked next is, well, okay, then how does this correspond to survival? If we look at our imaging results, how does a tumor's accumulation correspond to survival? So what we did is we drew regions of interest in the tumor, and that was the right hemisphere. So we did not draw it on the hot spot. And for blood, we drew the right ventricle or left ventricle. That was the region of interest to represent blood. And here is the definition of SUV, unitless value. And the way we work this is we determine the Pearson correlation coefficient between the SUV mean and days of survival. So over on the left, you can see at 24 hours, there's a fair amount of scatter there. But at later time points, the numbers coalesce into a high correlation coefficient. Now, most of the vehicle treated groups, all the mice were dead by day 25, and that's the place where we had the lowest SUV values in these treated groups. In this table, the Pearson correlation coefficients are all relatively high, but they don't become significant until about 48 hours. And this is consistent with what we know already about imaging with IgG, is that somewhere around 48, 72 hours, you get your best signal to noise ratios. The blood is on its way out of the tumor. So there isn't, we did everything at 144 hours, in other words, did everything. I meant the BioD. I think now that we have this data, we may go back and do some more optimization at earlier time points. So for a summary here now, we've shown that we can induce CD69 positive T cells in spleen and tumor, but with differing kinetics. There was greater tumor accumulation of the zirconium-89 anti-CD69 antibodies in the ICI treated group relative to the vehicle control group. And we also showed that the tumor accumulation corresponds to increased survival. So I think that now we should really go back and look at early time points that would warrant. I mean, the way this might play out in the clinic is that we could look at earlier time points to decide whether or not you could continue ICI treatment. Some of the things we're going to look at are antibody fragments. They clear faster. We could even perform serial imaging in the subject. I think we need to up our game and start co-registering the MRI tumors with PET and draw our regions of interest right on the tumor. I think we'll have more accurate quantification of the SUVs. All right, we're almost at the end. A lot of that work was done at the University of Pittsburgh in the in vivo imaging facility there, but we're looking forward to doing it right here at the Molecular Imaging and Theranostics Center, METC. It's just next door to Merck. Now, I gave a talk in August and had this slide up there, and at that time we thought that we might be opening in January, but looks like it's going to be May. And at that time, we're going to have a Bruker BioSpec 3T MRI preclinical scanner, in addition to a Bruker Albira PET, SPECT, and CT. So we're looking forward to doing our experiments on those. Can't wait. And as always, we thank our collaborators, our funding, a whole lot of people, hardworking people made all this work possible. So that's it. Glad to field some questions.
Dr. Edwards, thanks.
Hi.
That was a fantastic talk. Emma, go ahead.
Yes, so Barry, some of you know me, but I just want to introduce myself since I haven't had the pleasure to meet you. I am Emma Teixeira. I'm an immunologist, a T-cell immunologist actually at MMI and at NextGen, very much working on T-cell differentiation, all of the subtypes, multiple subtypes of T-cells that are involved in tumor clearance and how they are, our knowledge is evolving on them. I'm very much interested in your topic and great talk, by the way. So my question has to do with all of these antibodies that you guys are using are checked not to be agonist antibodies, right? Meaning that they don't activate the cells when you provide them.
That's an interesting question. And I do not know the answer, but what I would like to say is that the amount that we administer as a tracer should not, the amounts that we administer generally do not have agonistic function. And the amount of tracer that actually, makes it to the tumor site, it's something we need to think about, which we generally don't, because we're thinking of so many other things. But that is an important consideration to ask us whether we're altering the very target we're trying to measure.
Right. And then, so for T cells, for Ts, the field is advancing, so there is not an absolute marker for them. Usually they are the different subsets, they have a lot of different colors, right? I call it, so my question has to do is it possible to use combinatorial, cocktails of antibodies that are coupled with different radioisotopes, so it's not toxic, but you can somehow identify better the subsets of the Ts that are there. Like, for example, C69, it will target not only Ts, but also Tregs and other cells. So is there a possibility to use more than one antibody at a lower dose?
Well, we can do that, but we'd have to do, if it's going to be in the same animal, it would have to be done serially. We only have one color in PET. So there's not an opportunity to do that. The other way to do it, it would be to try one antibody that's a fast clear with a short-lived isotope, follow it up quickly with another antibody that clears a little bit more slowly, or also quickly with a short-lived isotope, like maybe CD68 antibody followed with another CD or gallium-68 antibody.
Yeah, the reason why I'm asking this is that some of the, and you may know this, but some of these subsets are more amenable to checkpoint blockade than others. And so that is important to distinguish actually in terms of, diagnostics or So then my last question has to do with what is your best isotype for half-life? How you have talked about, I think, 6 or 7 days. Can you use one that is longer than that?
Oh, interesting question. Well, we have to use what nature gives us. And for PET imaging, zirconium-89 is probably one of the longer, readily available half-lives out there. Lutetium-177 has a 6 or 7 day half-life. It just happens to be not a good isotope for any kind of imaging. And that brand of imaging would be SPECT.
Because I see that, so the fact that you guys are targeting the C69 positive cells that are resident memory has potential of application not only for cancer, but for infectious diseases, and being able to monitor them through time, it would be very important. So I'm thinking of either half-life or how often can you administer it so you can kind of track it.
Oh, right. Okay, great. Yeah, the solution to that would be probably use a shorter a radionuclide with a shorter half-life and just do serial imaging with antibody fragments, which clear faster.
Right. Well, thank you very much.
Oh, well, thank you. Thank you for your questions.
Further questions for Dr. Edwards? If not, I have a question to follow on to what Emma was asking. Have you considered pre-targeting strategies? So you could tag your T cells and then periodically use a short half-life tracer in a pre-targeted type chemical connection so that you could see them over several days after one antibody tag that circulated for a week or something like that.
Yeah, it's, it came up. I mean, my collaborator Gary wanted to do it, and that, at the time I was all thinking about patent technology and intellectual property and translation, and I had my doubts that pre-targeting is ever viable for commercial translation. Mm-hmm. But it's a good idea though in this context though to try. So thanks for bringing it up.
Yeah, we've managed to pre-target at least a bisphosphonate ligand that we think we can probably target multiple times in a dog. So we made that work. We'll see how we do as we keep moving forward with it.
Okay, well, that's a good idea. I'm going to rethink that because there's always important information to be teased out of preclinical models. And we could do that with the pre-targeting approach.
Yep, definitely. Other questions for Dr. Edwards?
I have the one question. Can you hear me?
Yes, Guanghu, go ahead.
Okay. So Barry, so you give a very good talk. I'm very interested in your study. My question is that when you use the CD11B as the target for the macrophage, so the CD11B definitely is the general is the typical markers for all the different macrophages there. But for the tumor growth, usually we think about the tumor-associated macrophage. So that's have some another kind of marker such as CD206, probably more useful for the therapy there. So do you have the opportunity to use another marker such as CD206 to do some actually? So this nuclei therapy use your strategy?
Well, we're actually undertaking those experiments right now at the University of Pittsburgh, targeting CD206 with for alpha therapy. And then just the reason that we went the route that we did was really a grantsmanship approach because CD206 had already been done. And we knew we weren't going to get that funded.
Okay, thank you.
Well, Barry, that was a fantastic lecture. Really thoroughly enjoyed that. Thank you so much for joining us today. And we've used just about exactly the hour, so perfect timing. Thank you so much for what you shared with us.
Oh, glad to do it. Looking forward to the next one in a few years.
Sounds good. And thanks everyone for attending. This is the attendance is growing and we look forward to our in-person meetings down the road. I think the next couple are still on Zoom and hopefully we'll be in the NextGen building in future meetings starting in the summer.